molecules are the coenzyme NADH and NADPH, which are found in abundant millimolar type concentrations within cells. And another molecule called glutathione, which I'm going to say only once. It's difficult to say for me. And it's a peptide, a weird peptide with an atypical peptide bond for its first glutamic acid residue. But importantly, it has a cysteine. And you remember cysteine can form disulfide bonds. But it can also donate a hydrogen and reduce another cysteine. All cells, almost without exception, have this peptide, and they have a system to regenerate it, to keep this cysteine reduced as a thiol group, SH, so that this thiol group can donate its hydrogen to cysteines in proteins, keeping those proteins reduced, free thiols. On the other hand, proteins, etc., that live on the outside of the cytosol often have disulfide bonds. They are not reduced by the action of this peptide. And so disulfide bonds there are common. Why? Because the chemistry is unfriendly, and because these proteins are not in the protected environment of the cytosol, and they have to deal with conditions on their own. Think about the snake venom proteins and the digestive enzymes. Now also, presently, on the outside there's oxygen, and the environment is oxidizing, which favors the formation of disulfide bonds if it wouldn't be prevented by this molecule. So there's a fundamental difference, but not all. We also see differences in that proteins that are inside cells are not so tightly folded. That is not the most important thing for them, because they need to be active. But they often associate together in large multi-subunit complexes. And there are various examples of that in the TCA cycle and in other parts of metabolism. And the whole ribosome is an example of that. So these proteins are often in big complexes working together, whereas the proteins that are on the outside of the cell are often smaller, compact, single subunit proteins that fend for themselves and that have, once again, a lot of disulfide bonds. And all of these proteins that I'm listing here, RNAs and lysozyme, that's an enzyme that we produce in our tears, which has antimicrobial function because it degrades the peptidoglycan of bacteria, or proteins that function in respiration and photosynthesis, but that are in these exocytoplasmic compartments are always small and compact. And these are very stable proteins, which is why they are studied so well. Now, because of all of this, these enzymes and proteins that function outside of the cytoplasm can tolerate more aggressive conditions. Inside the cell, aggressive conditions are prevented. This is the whole idea of having membranes. You have a peroxisome because you keep the nasty chemicals in one place. That's an idea. Why do you want to do that? Because in the cytosol you have what the cell values most, and that's its genetic material, DNA and RNA. This is not present on the outside of the cell. There's no function there. There's no protein synthesis there. All the proteins are synthesized in cytoplasm or cytoplasmic-like compartments. I believe these are fundamental, important differences. We can ask the question, how are these differences achieved and how is it maintained? And that's what we'll look at in section 2, where I'll start very soon. Now, I'd like you to, one more time, review the structure of eukaryotic cells as it's depicted, for instance, in figures 1.8 and 1.10 in the book. You really just need to know the main organelles. What is part of the endomembrane system? What is a Golgi apparatus? What is the ER? What is the peroxisome? On and on. So that you recognize the overall structure, you have some idea of the basic function of these. How are we doing? The point I just tried to make is for me really important. And there is biotechnological application also to this idea. So let's say you want to express a protein in a bacterium because a biotech company pays you $10 million to do that. This is not an imaginary problem, by the way. If your protein if it's a human protein, and in human cells, this protein will be active in the cytosol, you are going to express it within the bacterial cytosol. That's the condition that matches. But if it's a protein that is active in a lysosome, in the human cell, then you would secrete it from the bacteria, because then it will find itself in an environment where it will probably function and fold in the same way as it did in the human cell. I think this is important. Enough about that. All right. Woohoo, that was section 1. Section 1, the components of the cell, how cells are studied. We've been there. Now, we'll look at section 2. And section 2 is cells in action. I like that. Cells in action. Action is good. Okay. So what types of action do cells have? Well, cells have all kinds of action, but an important type of action is transport functions. It would be perhaps a surprise to you, but about a third of the ATP that we spend in every cell in our body is used to move things across membranes. About a third of the ATP. And if you go to neurons, it's the majority of the ATP. It's like 76, 60 to 70%. So membrane transport is an important part of the action. Now let's look at membrane transport processes. And what we will do before the exam is we'll look at individual transport processes, and then we'll look at how that comes together. So how do cells use transporters to together form a system? And then ion gradients, etc. We'll look at that after exam 1. Okay, so here we go. How can we study transport? Well, we need a system to study, an experimental system. And we can have cells that we can put in a tube, and we can add chemicals. And if we can measure the chemical, we can measure if it gets into the cell, yes or no, by measuring changes in the concentration on the outside or inside the cells. Or we can have other systems where we isolate membrane-forming lipids, which by the way you do by adding chloroform and methanol to cells. You extract the lipids, you can then separate them using chromatography, and then you can put them back in water and you can spontaneously these structures, which we call liposomes. Now, if you've also purified a protein that you think is a transporter, not an easy task, but if you've done that, you can incorporate that protein in your liposome. So, you have your liposomes, small vesicles, or vesicles, as the British say, or your cells, you put them in your swimming pool, somewhere in a tube. You add your molecule X, and you collect your vesicles or cells by centrifugation, and if X made it into the cell, you can measure that. So we measure that at a certain concentration of X, and we see the uptake. And from the slope here, we get the rate. Now we do this again, but we change the concentration of the molecule X, the substrate concentration, we change it. Do it again. Measure the uptake, we get the slope there. We have the amount of molecule taken up. It's a function of time. The slope here is the uptake rate, now at a second concentration. And we repeat this at another concentration on and on. In the end, we have measured at a different set of concentrations the rate of uptake. So we have concentration on the horizontal axis, and we measure the rate of uptake. And we plot that, so you get this type of curve. And you recognize this curve because it looks like a Michaelis-Menten plot. So this is what we call a Michaelis-Menten plot. not for an enzyme, but now for a transport system. So we can do that. So why do we want to do this? We want to do this because what a curve looks like tells us a lot about what the type of transport mechanism could be. If we have a very slow rate of uptake, but it's going up linearly with the concentration, then we suspect that this molecule is just diffusing into the cell passively. We call that this first pathway, passive diffusion, or simple diffusion, where the molecule moves from a higher to a lower concentration, such as oxygen moving into the blood cells in the lungs. And later, in the peripheral tissues, oxygen moving out of the blood cells into the tissue. So oxygen diffuses passively. On the other hand, we can have a very rapid rate of uptake, but we never saturate it. So then we think we maybe have a hole in the membrane that specifically allows this molecule to get in. So that's a channel function. So the molecule, in this case B, goes from a higher to a lower concentration and then makes it into that compartment. So an example of this is sodium channels. As I said, sodium is always high on the outside of cells, low on the inside. So if you have a channel in your membrane, the sodium will move from the outside into the cell. And it can be a fast rate because there's nothing stopping that sodium. We don't need to bind it. On the other hand, we can have this curve where there seems to be a saturation. And we go up and up, the rate goes up, and at some point the rate stops. This is because there's, like in enzymes, a binding pocket in the transporter. And there are two types of proteins that could have this function. We call these the carriers or transporters. So we have a molecule that binds to the transporter and gets through. Glucose, for instance, gets into our cells by this type of mechanism. But another possibility is that we have a molecule that uses the energy stored in ATP to move something else across the membrane. This is called a pump. So pumps on the B, carriers on the C here, are types of transporter proteins that specifically bind the molecule. You can think about the saturation mechanism by thinking about a river, and you have a small boat that can be taken across. And if you just have a couple of people on one side of the river and they all fit in the boat, then the whole group goes across pretty easily. But if you get a big group and a small boat, then you get a certain maximum rate of transport by which people can go across the river. As if you're loading capacity for the seats in the boat, and then with more passengers you can move more people across as long as you have the loading capacity. Similar to enzymes, so there's a saturation for some transports. So the type of curve that you get tells you a lot about the transport pathway. Okay, so let's first look at diffusion. So diffusion, remember, we suspect that we have diffusion if we get a relatively slow rate of transport, and the rate of transport is linear with the concentration. So oxygen is an example. It moves by simple diffusion. Now, if we study diffusion in detail, we see that what really matters is the ratio of concentrations on the two sides of the membrane. So it's the concentration difference. So the rate of diffusion or the flux is really proportional to a constant and the concentration difference. So the bigger the concentration difference, the faster things diffuse. What affects the constant? Things move more easily through a membrane if they make less hydrogen bonds with water. So molecules that make a lot of hydrogen bonds with water transport really poorly across membranes. Think about glucose. There are 6 oxygens in there. All those oxygens want to be hydrogen bonded with water. There's no way by which glucose will diffuse. But if you think about something like ethanol with one hydroxyl group, yes, that wants the hydrogen bond with water, but not that much for the overall molecule, so ethanol diffuses pretty rapidly. If you make longer alcohols with longer acyl chains, they diffuse even more rapidly. Oxygen, is a little bit polar, of course, but there's not a ton of hydrogen bonding that oxygen can do, and the oxygen concentrations are so high that we still see appreciable transport rates. Now, a special case of diffusion is the diffusion of water. And per water molecule, the probability of it diffusing across the membrane is very low. But the water concentration is always close to 55 molar, it's very high. So the net rates of water diffusion are actually appreciable. And remember, water diffuses in the direction of where there is the highest solute concentration, and that corresponds to the relative lower molar concentration. So we call this process osmosis, and what we need to remember, animal cells like to be in a solution where the amount of solute on the inside of the cell and on the outside of the cell is in equilibrium, is about the same. So the amount of dissolved particles inside and outside is about the same. That means there's the rate of diffusion in and out of water is the same, so the net osmosis is zero. So they're not shrinking as they will do in a solution with excess solvent, which is hypertonic. Or they're not swelling in a solution with too low amounts of solvent, which is hypotonic. Plants, on the other hand, because they have a cell wall, they like to take up water, only their vacuole that puts pressure against the cell wall, and that keeps that cell turgid, keeps that plant upright. So plants like it hypertonic. Okay, and that's enough for today. We'll look on Friday at transporters and Monday at transport systems.